Hello students, welcome to my class. So in this video, I will be starting with unit 2 of your bioinorganic chemistry paper, that is transport and storage of dioxygen. So here in this topic, we'll be studying about how oxygen is transported in the body of various mammals or different organism, how it is being stored, what are the different types of proteins that are involved in this process. Okay, so to begin with, we are studying about oxygen carriers. So what are oxygen carriers? As you can see, the name says that oxygen carriers, meaning to say they carry or transport oxygen in the body of maybe any organism. Okay, so they are special type of metalloproteins, and what do they contain? They have transition metals from the 3D series that are present, that are bound to the protein. So these are a special type of metalloprotein. Metalloprotein meaning to say they have a metal in them. That is the transition metals from the 3D series. Only transition metal can form dioxygen complex with O2. That is the reason there can't be any other metal from other series. There should be only transition metals from the 3D series, because only they have the capacity to form a bond, that is a complex with oxygen molecule. What is the basic requirement of an oxygen carrier? It should carry oxygen, correct? In order to carry oxygen from one place to another, it should form bond with oxygen, no, first. Only then it can take it and carry. So in order to form bond, it should form a dioxygen complex, and only these transition metals can do that because they have a vacant site for binding and they exist in higher oxidation state. So they themselves won't get oxidized because they are already in the higher oxidation state, and they have a vacant site wherein they can bind and occupy oxygen and then take it. So they prefer transition metal in the oxygen carriers. And the common examples of these transition metals can be iron, copper, or vanadium, which is present along with the protein as a metalloprotein, which is nothing but your oxygen carrier. What is its function? To carry oxygen. So next, a very important question, that is, what is the basic requirements of an effective oxygen carrier? Now, if I want a good oxygen carrier, what are the requirements that it should have? First thing is, a good oxygen carrier should bind to oxygen reversibly with optimal affinity so that it can release the bound dioxygen to an acceptor at a rapid rate. Meaning to say that this oxygen carrier, it should bind to oxygen in such a way that it is not bound very strongly also, and it is not bound very loosely also. One optimal affinity in between, so that whenever it reaches the receptor site. Now if we're talking about human body, you know that oxygen is carried from the lungs to your muscles where there is requirement of oxygen. So the receptor site is your acceptor site is your muscle, okay, and the receiver site is your lungs, correct? So this oxygen carrier should be bound to that oxygen in such a way that it is able to go and release it in the muscle, and it should not be like very loosely bound also, so that it doesn't lose the oxygen on the way when it is taking it to the location or to the muscle. So that optimal affinity it should have, that is one requirement. Second thing is functioning of a dioxygen complex. Now this is a complex, dioxygen complex, LNMSO2. Here what is L, students? That is a ligand. You know that in any complex there is ligands. What are ligands? They donate electron pairs to the central metal atom. So L is a ligand, M is a metal. It may be iron, copper, or vanadium, any transition metal. S is here the substrate, which is supposed to be oxidized by the oxygen. Okay, any substrate. So the functioning of this dioxide complex as an oxidase enzyme or oxygen carrier, it depends upon the quantity of two important constant, that is big K and small k. Two rate constant, big K and small k. Now what is this? So you should be able to write this reaction, students. What is this? This is a dioxygen complex. Then what does it do? It forms a coordination bond with oxygen, correct? So there is bond formation of this substrate with oxygen. Now what happens, which is supposed to be oxidized here? The substrate S is supposed to be oxidized. So next it gets oxidized and this complex breaks into LNM. So its work is over. The ligand and the metal complex work is over. It is supposed to bind to oxygen and then oxidize this substrate. So it oxidizes the substrate. Now S is in the oxidized form. What happens to oxygen? Because it oxidized another species, it itself gets reduced and the ligand metal is released. So you can see two rate constants are there, that is big K and small k. In order for this to be a very good effective oxygen carrier, the K value, that is this K should be large and this K should be small. Okay, that is rate constant value. The first rate constant should be large and the second rate constant should be small. Only then this can be considered as an effective oxygen carrier. So what are the two properties once again, students? One thing is it should be bound to oxygen in an optimal affinity so that it can release it whenever it is required and it can take up oxygen whenever it is required. And the second property is you should know the equation and how the two rate constant values should be there. The first rate constant value should be large and the second rate constant value should be small. Next, we are starting to discuss about an important oxygen and storage type of protein, that is hemoglobin, which is an oxygen carrier, and myoglobin, which is an oxygen storage protein. So hemoglobin, you can find it in human body. Other than that, birds, fish, reptiles, and some insects. Myoglobin also is seen in human body, birds, fish, reptiles, and other insects. And what is the transition metal present in both of them? That is iron in Fe+2 oxidation state. It is seen in both hemoglobin and myoglobin. And hemoglobin, what does it do? Its function is it functions as a carrier type of protein, whereas myoglobin, it functions as a storage type of protein. What is the difference? A carrier is something that carries oxygen from one place to another, from maybe the giving side to the receiving side, whereas a storage type of protein, it stores oxygen in itself until it is required for some reaction. Okay, it does not carry, it only stores. Whereas carrier type, what it does, it is like transporting from one place to another, it drops the oxygen. Okay, so in deoxy form hemoglobin, that is deoxy form means when hemoglobin does not have oxygen, or when it is not bound to oxygen, it is red purple in color, and when it is bound to oxygen, it is red in color. So now let us study about both hemoglobin and myoglobin. What is their structure and function? Okay. So hemoglobin is an iron containing oxygen transport metalloprotein belonging to the family heme protein found inside the red blood cells. Okay. You know that hemoglobin is present in the RBC of our body of almost all vertebrates and as plasma of some invertebrates. Plasma is the liquid that is present in invertebrates like blood only. So it is present in the plasma of some invertebrates and in the blood of almost all the vertebrates. So what is the metal it has? Transition metal is the iron. And what is its work? It's a metalloprotein which is used in oxygen transport. So hemoglobin carries oxygen from lungs to the tissues where it is transferred to the myoglobin. So what is hemoglobin doing? It's taking the oxygen that's come into lungs to all the tissues and muscles, where then in the muscles there is myoglobin. This hemoglobin transfers that oxygen to myoglobin and it is stored there for further metabolic requirement. Whenever the muscles need it, it is being stored in myoglobin and then it will be provided to the muscles by the myoglobin whenever it is required. So each red blood cell, it contains around 300 million hemoglobin proteins, and these are responsible for carrying oxygen from where? From the respiratory organs like lungs, or if it is in case of fish, it is gills, to rest of the body parts and the tissues. So coming to the structure of hemoglobin and myoglobin, we can study this both together because both have almost a similar structure. Only in case of hemoglobin and myoglobin, difference is that myoglobin is a monomer, whereas hemoglobin has the same thing, same structure four times. That is, it is a tetramer. Okay, that we'll study further. So we'll begin with myoglobin. You should be able to draw the structure. This is the structure of heme unit in the hemoglobin and myoglobin. So this part constitutes the heme part, and then this is a protein part, that is a globin chain. Okay, uh, that is the proximal histidine and the distal histidine and the globin chain, and this part constitutes the heme part. So myoglobin, they are relatively small protein that consist of 150 amino acids, and they're considerably smaller compared to that of the hemoglobin. And functional unit of myoglobin is an iron porphyrin complex that is embedded in the protein. That is this one. This is the functional part of the myoglobin, which is the iron. And what is this ring? You also studied this in chlorophyll. Correct. This is a porphyrin ring. So iron, this time there it was magnesium. Now we have iron that is present in the as a central metal atom in the cavity of the porphyrin ring. So this is the functional unit of the myoglobin. In myoglobin, the heme iron is 5 coordinate with only single histidine imidazole ligand from the protein. That is, this is the heme part, okay, of the hemoglobin as well as myoglobin. This is the heme part, and it has the fifth coordination. You can see four coordinations of the iron are satisfied by the four nitrogens from the porphyrin ring, and the fifth coordination, it is satisfied by proximal histidine. Why is it called proximal histidine? Is because it is present close, in close proximity to iron atom, so it is called as a proximal histidine. So four coordinations, they are satisfied by nitrogen, and the fifth coordination here is satisfied by this proximal histidine of the globin chain. And then there is a distal histidine. That is, what are these histidine chains actually? They contain imidazole ring. What is an imidazole ring? It has two nitrogen atoms. Can you see? So this is a proximal histidine. This is a distal histidine. So it cannot form a bond with this iron. Why is that, students? Because it is very far away from this iron center. Because it is too far away from the iron to be bound to it. Hence, distal histidine, it does not form a bond with the central iron atom. Reason is because it is too far away. Whereas proximal histidine, the imidazole ring of the proximal histidine can form a bond with iron because it is close enough. So, how many bonds does this iron have now? 1, 2, 3, 4. 4 coordination. They are satisfied by the nitrogen of the porphyrin ring. Fifth coordination is satisfied by the nitrogen of the proximal histidine. Sixth coordination, consequently, the iron atom has a vacant coordination site. That is, its sixth coordination is not satisfied by anybody. So, it has a vacant coordination site. This is where the oxygen binds. Now, myoglobin, you know that oxygen storage protein it is. So, whenever hemoglobin is coming and giving oxygen to this myoglobin molecule, it should bind to that oxygen. Now, where does it bind? This iron has a vacant sixth coordination, and that is where that sixth coordination binds. Okay. So, what is the structure of myoglobin did we study? It has a non-protein part that is called as a heme unit. That is this part. And then it has a protein part which consists of the proximal histidine, the globin chain, and the distal histidine. And then there is a central atom, that is a transition metal, which is iron. Iron in +2 state is present as a central metal atom. Okay, and then the four coordinations of the iron, they are satisfied by the nitrogen of this porphyrin ring. And the fifth coordination is satisfied by the imidazole, which acts as a proximal histidine because it is present closer to this central metal atom. And whereas the sixth coordination is actually vacant, because of which oxygen that is coming can bind to that sixth coordination of the ion. And distal histidine, it is present far away from the central metal atom. It is also an imidazole ring, but it is called distal histidine because it is present far away from the Fe center, so it cannot form a bond with Fe. So this is the heme part, and this is the heme part, which is a non-protein part, and this is a protein part or the globin chain. Now, one thing you have to study is that in deoxymyoglobin, that is when sixth coordination is not satisfied, the ferrous ion, the fifth coordination is not satisfied, so it is in a high spin state. So because of this, Fe2 plus, when it is present in high spin state, it is little bit large in size, and it cannot fit into the hole of the porphyrin ring. So it is present 50 picometers above the porphyrin plane. That is, this Fe plus 2, whenever this myoglobin is in the deoxy form, what happens? It does not have a sixth coordination. Oxygen is not there in the sixth coordination site, because of which Fe plus 2 has a bigger size, and because of that it cannot fit into the center of the porphyrin ring, and because of that it is present around 60 picometers above this center plane of the porphyrin ring. But what happens in oxyform? Whenever oxygen binds to that myoglobin, the deoxymyoglobin gets converted into oxymyoglobin. So the sixth coordination is now satisfied, and the Fe plus 2 ion is said to be in low spin state. That time it was in high spin state because its sixth coordination was not satisfied. Now it has come to lower spin state because the sixth coordination is satisfied by oxygen atom that is bound to it, and now it becomes comparatively smaller in size, and it will fit properly into this cavity of the porphyrin ring. That is the difference between deoxymyoglobin and oxymyoglobin. In deoxymyoglobin, you can see this Fe plus 2 around 60 picometers above the plane, whereas in oxy form it is in low spin state, so it exactly fits into the center of the porphyrin ring. Okay. Next, a heme unit including globin protein chain is called as a myoglobin. Now you understood from the structure that one heme unit containing the globin chain is nothing but your myoglobin. And it has a molecular weight of around 16,000 Dalton. Now, what is the difference between hemoglobin and myoglobin? Hemoglobin, it has a molecular weight of around 64,000 Dalton. It is a tetramer of myoglobin unit. That is, myoglobin's 4 times is nothing but hemoglobin. Okay, so these 4 units, these same units are present in hemoglobin, but they are present 4 in number. In myoglobin, it is only 1, whereas in hemoglobin, they are 4 in number. So those 4 are similar, but they are not identical. So you can see here, this is the difference between this myoglobin structure and this is the structure of hemoglobin. Now you could see that central ion is there here, and that globin chain which is folded now. There we showed it in the form of molecular structure, but actually it is present like this. That globin chain is folded, and this is the heme part, and this is a proximal histidine. Okay, and distal histidine will also be there somewhere over here. So only one unit is present means then it is called as a monomer, which is a myoglobin. Here you can see 4 of it is present. Then it is a quaternary structure, that is a hemoglobin. You can see there are 4 such units that are present, and there is high folding of the globin chain, and these are the 4 Fe present in each of the subunits. So this is the hemoglobin structure. Now let us study about hemoglobin. So hemoglobin, it is considered to be a conjugated protein, where same way like in myoglobin, same points you can write over here also. That is, heme is a non-protein part and globin is a protein part. Now what is the difference? That was a monomer. This is a tetramer with a molecular weight of around 65,000 Dalton, and it consists of 4 homologues, that is same looking globin chains. These are connected together by disulfide linkage. You can see that each of it has this globin chain, and these all the 4 globin chains of different units, 4 units, are linked together by disulfide linkage. Next is a normal adult hemoglobin consists of two alpha globin chain comprising of 141 amino acids and two beta globin chain. It has 146 amino acids. So in case of adults, it is two alpha and two beta globin chain. Whereas in case of infants, that is small kids that are just born, they have two alpha chains and two gamma chains. That is the difference between adult hemoglobin and infant hemoglobin, where adults have two alpha and two beta globin chains. The infants, they have two alpha and two gamma chains. Once they grow up, the gamma chains are gradually replaced by beta chains. As they grow up to be adults, their gamma protein chains are replaced by beta chains, and the arrangement of these amino acid residues present in the protein, they are responsible for the shape and the function of the protein, that is this hemoglobin. How these chains are arranged together, how they interact, they are responsible for the structure and the function of this protein chain. So next is the structure of hemoglobin. You can write the same points that you have studied under myoglobin over here also, because it is the same structure, correct? Only difference is this is a tetramer. So now we are studying about one unit, that is one monomer of that tetrameric structure of hemoglobin. So attached to the globin chain is an iron porphyrin complex. This is called as a heme, correct? We already studied it over there also. And its role is transporting oxygen and carbon dioxide in our blood. That is, it takes oxygen from the lungs to the tissues, and the carbon dioxide that's given out from the tissues is carried back to the lungs. And then the heme group, it contains iron atom that is present in the center of the porphyrin ring, and there the iron is coordinated to four nitrogen atoms of the porphyrin ring. Who satisfies the fifth coordination? The fifth coordination, it is satisfied by the imidazole ring of the proximal histidine of the protein chain. Okay, so the fifth coordination is satisfied by this imidazole ring of proximal histidine, and the sixth coordination site is vacant, where it binds to oxygen in the oxyhemoglobin and also water. Okay, so the sixth coordination is bound to either oxygen or water. In this is in case of oxyhemoglobin. What happens in deoxyhemoglobin? The sixth coordination remains unsatisfied, that is, nothing binds in the sixth coordination site. And also you know that there is a distal histidine that is present, that is near the sixth coordination site. There is another distal histidine, and because it is far away, it cannot form a bond with the central metal atom, but still it plays a very important role. Now, this is an important question. What is the role of distal histidine in the structure of hemoglobin? Distal histidine has two important roles. One is it stabilizes the oxy form of the hemoglobin through hydrogen bonding. So, whenever hemoglobin is bound to oxygen, then it is called as a oxy form of the hemoglobin. That form is stabilized through hydrogen bonding by the distal histidine. One more function is it protects the hemoglobin and myoglobin from CO poisoning. Okay. So it protects it from carbon monoxide poisoning. Also, it stabilizes the structure through hydrogen bonding. And each hemoglobin binds to four oxygen molecule. Why is that? Because each hemoglobin has four such monomer units having four iron atoms. So it is each heme unit is able to bind to one oxygen. So when it is a tetramer, so it is able to bind to four oxygen atom because it has four such units. So each hemoglobin, it binds to four oxygen molecules since it contains four iron atoms, and each of it is able to bind to one. And uh, it is iron contained in this hemoglobin that gives the red color to our blood. So you have seen that our blood is red in color, and usually you have heard that it is because of the hemoglobin pigment that is present in the blood, your blood has red color, but it is actually because of this iron that is present as a central metal atom in the hemoglobin, in the heme protein. It is because of that the blood is red in color. So in hemoglobin, you know that there are four heme units, and hence each heme unit has a globin chain, which is a polypeptide chain. So these four polypeptide chain, they are coiled in such a manner that these four heme units, they are present towards the corner of the tetrahedron near the surface of the molecule. So you can see that these four units, the heme units, are there in such a way that it appears like a tetrahedron. These are present in the corners, and these chain, the protein chains, they are coiled with one another. So these protein chains, they have this different ions in them, that is CO2 minus or NH3 plus groups that are present in the chain, that interact with one another. Such interactions of the ions that are present in the globin chains, the protein chain, is called as salt bridge interaction. That is, these NH plus groups interact with the CO2 minus or different groups. Such interaction is called as salt bridge interaction. And one more thing you have to know is that each of this heme unit has one DPG molecule along with it. What is DPG? It is 2,3 diphosphoglycerate molecule. So each heme unit will have one such DPG molecule. So these are the four heme centers, and these are the globin chains. Okay, where you can see the charges. And there is one DPG molecule. So what happens? DPG, it has a large number of negatively charged sites. That is, you can see that it has large number of negative charged sites which interact with the positively charged residues of the chain. That is the NH3 plus of the beta chain. Because of all these interaction, it is responsible to give a quaternary structure to the deoxyhemoglobin, and that structure is termed as the tensed form or T form of the hemoglobin, of the deoxyhemoglobin. Why is it called as a tensed or T form? Because there is lot of salt bridge interactions between the negatively charged ions of the DPG and the positively charged residues of the beta chain of the hemoglobin. Whereas whenever it is oxygenated, in oxygenated form, what happens? All these interactions are destroyed. There is no salt bridge interaction seen in oxygenated form, whereas in deoxygenated form, these interactions, they exist. So in oxygenated form, it is said to be in oxyhemoglobin is said to be in relaxed form or R form, whereas in deoxy form it is said to be in tensed form or T form. Why is it that? Because in tensed form it has salt bridge interaction, whereas in relaxed form it is oxygenated. As soon as it gets oxygenated, the salt bridge interactions are destroyed completely. There is no salt bridge interaction. And last thing you have to know is that this is the same thing you studied in myoglobin. In deoxy form, what happens? Fe2 plus is in a high spin state and it is bigger in size. So it is present 50 picometers above the porphyrin ring. Whereas in oxyhemoglobin, what happens? Fe2 plus will be in low spin state and its size will be smaller. Hence, it will fit into the cavity of the porphyrin ring. So next is the characteristics of O2 binding interaction with hemoglobin and myoglobin. This is another very important topic. So the first characteristic we are going to study about is about cooperative effect, Hill point, and allosteric effects. Okay. So in simple words, if I have to tell you what is cooperative effect. You know that one hemoglobin molecule can bind to how many oxygen atoms? It can bind to 4 oxygen atoms. What is cooperative effect? So as soon as it won't bind to all four at once, it is like one by one it will bind. So as soon as one hemoglobin molecule binds to one oxygen atom, what happens? The protein conformation changes. That is, its structure changes, and because of this changes in the structure, it influences or it prefers to bind to the next oxygen. It makes the binding of the next oxygen easier. Okay. Are you understanding? One oxygen binds, and so the structure of this hemoglobin molecule completely changes, conformation changes, which influences or which helps in binding of the second oxygen molecule. It prefers to bind to more oxygen. Same way, when it binds to second oxygen, same way it prefers to bind to one more oxygen, like that. So oxygen affinity goes on increasing, preference to bind to oxygen goes on increasing, because as soon as one oxygen binds, it prefers to bind to one more oxygen. It is like, yeah, you are joining one group. Say you joined some fun activity like a group, and as soon as you joined, you are helping your friend to get in. You joined a group and you're finding it really good. Okay, so you want your friend also to join. So what will you do? Your affinity to join your friend has increased, so you will join your friend next. Once your one friend has joined, once again now you two people are there who are very interested in the group. Now you want your one more friend to join, so you are bringing one more friend into it. Once you are three people, now again your affinity to join one more friend has increased, so you're bringing one more friend, like that. Okay, so oxygen affinity is increasing. So what is, as one oxygen binds, the conformation of the protein changes, which again favors the binding of the second oxygen. So binding of the simultaneous second and third and fourth oxygen is favored by binding of one oxygen. Because of binding of two oxygens, the third oxygen binding is favored. Because of binding of three oxygen, the fourth oxygen binding is favored. Now after this it won't continue. Why? Because the maximum oxygen it can take up, hemoglobin molecule can take up, is 4. One hemoglobin molecule can take up maximum of 4. That is nothing but your cooperative effect. Okay, due to positive cooperativity among the subunits of the hemoglobin, successive oxygen binding constant. What is this K? Oxygen binding constant. It increases in the order K4, K3, K2 and K1. This is like already three friends are there. They're trying to bring one more person, so that three of you will convince that one more person. Correct. Initially what, you are only one person and you're trying to convince the other person to come and join the group. Whereas when you reach 3 or 3 in number, 3 of you are convincing the other friend to come and join. So the oxygen binding constant, it increases. This is highest binding constant, because here 4 atoms are there. 4 atoms. Here 3 atoms of oxygen is there, so it is comparatively lesser than this, but more than K2. Then comes K2 and K1. This is the increasing order of oxygen, successive oxygen binding constants. So this is cooperative effect. So, can you define cooperative interaction? It is where the binding of one molecule of a substance influences the binding of next molecule of the same kind, and it is described as homotrophic allosteric interaction. Why homotrophic? Same type of atoms get bonded next. One oxygen is binding, it is trying to bind the next oxygen only, not any other atom. So same. So homotrophic allosteric interaction. Whereas this interaction is only seen in hemoglobin, but this type of interaction, it is absent in myoglobin molecule. So next, you know that what is the work of hemoglobin? It takes this oxygen from the lungs and takes it to the tissue. So in lungs, you know that oxygen pressure is very high, correct? Because oxygen is present more in number in your lungs. Whereas in your tissues or in your muscles, the oxygen pressure is low. Why? Because there is oxygen shortage there. Where is high amount of oxygen present? In your lungs. And in tissues, oxygen amount is less. So what should the hemoglobin be able to do? In the lungs, it should be able to bind to oxygen, whereas when it reaches the tissues, it should be able to leave that oxygen, because it cannot hold on to that oxygen in the tissue. It is supposed to drop off that oxygen in the tissues, whereas the myoglobin should take up that oxygen which this hemoglobin gives out. Correct. For this to occur, how does this happen in your body actually? How does hemoglobin take up? It is not like a vehicle, correct? It should work in certain way. So how does hemoglobin take up oxygen from the lungs? How does it bind to the oxygen in the lungs, and why does it leave this oxygen in the tissues? So this also can be explained from the cooperative effect. You know that in lungs, as I said, the oxygen pressure is high, correct? Meaning to say there is large number of oxygen molecule in the lungs, because of which there is cooperative effect shown in the lungs. Whereas is there cooperative effect in the tissues? No, the number of oxygen molecule is less, so that cooperative effect is not seen in the tissues. Hemoglobin carries oxygen from the lungs to the tissues where it is transferred to the myoglobin. To make this possible, this process thermodynamically possible, you know that oxygen affinity of the hemoglobin in the lung, where the oxygen concentration is very high, should be greater. That is what I said. And the reverse condition should happen in the tissues where oxygen concentration is very less. And the condition it is attained, our body is designed, hemoglobin is designed in such a way that this happens automatically. The oxygen binding curve explains this. So this is the percentage oxygenation and this is the oxygen pressure. Okay, so you can see that the oxygen pressure, when the pressure is low, meaning to say when the oxygen content is very less, percentage oxygenation, this is the curve for myoglobin and this is for hemoglobin. So when the amount of oxygen present is very low, that is pressure of oxygen is very low, percentage oxygenation of myoglobin is high whereas that of hemoglobin is low. Whereas when the percentage of oxygen is very high, that is when you come here the percentage of oxygen becomes very high, correct? That time oxygenation of myoglobin becomes less whereas that of hemoglobin becomes more. So you can see that for the myoglobin the curve is hyperbolic. This is a hyperbolic curve, that is going from high to low, whereas for hemoglobin the curve is sigmoidal in nature. The O2 binding curve can be explained by the cooperative interaction among the four heme units and non-cooperative O2 binding with the heme unit of the myoglobin. So you know that cooperative effect is seen only by hemoglobin and myoglobin does not show any cooperative effect. The reason why this curve occurs in this way is because of the same reason. This has cooperativity, this has non-cooperativity. Because of cooperative effect, whenever the amount of oxygen becomes very high, that time there is positive cooperativity shown. So the percentage oxygenation is also increasing of the hemoglobin. But since myoglobin does not show cooperative effect, when the amount of oxygen becomes very high, the percentage of oxygenation of myoglobin is becoming low. The oxygen affinity, how to measure the oxygen affinity, can be measured by P50, which gives the partial pressure of oxygen at which 50% oxygenation is attained. So you need to know the P50 value for myoglobin and hemoglobin. P50 value of myoglobin is very less, that is 1 torr. This is nothing but how much oxygenation, percentage of oxygenation that is taking place when 50, the partial pressure of oxygen when 50% oxygenation is attained. Okay, so for myoglobin the P50 is 1 torr and for hemoglobin it is 26 torr. Okay, very high value. What is this telling you actually? When the oxygen pressure is high or pressure is more, only hemoglobin will be oxygenated and the reverse happens in the muscle. What is the reverse? When the oxygen pressure is low, only myoglobin will be oxygenated. So what you are studying from all these 4 points, what is it telling you? You need to know to draw this graph. It is telling you that when the oxygen, this last point is telling you the whole thing, summarizing the whole thing. When the oxygen pressure is very high, who gets oxygenated? Hemoglobin. What is the reason for hemoglobin to get oxygenated? That is cooperative effect. Because oxygen pressure is high, meaning to say more number of oxygen atom. More number of oxygen atom means cooperative effect is high. When oxygen pressure is low, that happens where? In the muscle. Oxygen pressure is low means what? Number of oxygen atoms is less. That time it is the myoglobin which gets oxygenated more compared to that of the hemoglobin, because myoglobin does not show any cooperative effect. And P50 value for myoglobin is 1 torr, whereas that of hemoglobin is 26 torr. What is this value telling you? That at high oxygen pressure, hemoglobin gets oxygenated. That happens in the lungs. And at low oxygen pressure, myoglobin gets oxygenated. That happens in the tissues. So next is Hill coefficient, which is a continuation of the same topic. Hill coefficient, it is obtained from Hill equation. So you don't need to know the derivation how you get the Hill equation. This is the Hill equation, and N here stands for the Hill coefficient. What is Hill coefficient value telling you? One thing you have to know that if the Hill coefficient value is very high, it says that cooperativity effect is high. Okay. Whereas if the Hill coefficient value is very low, it is telling that there is low cooperative effect or there is no cooperative effect at all. So you know that hemoglobin shows cooperativity or cooperative effect, whereas myoglobin shows no cooperative effect. So for myoglobin, the Hill coefficient value n is equal to 1. That is very less. So it is telling us that myoglobin does not show cooperative effect, whereas for hemoglobin it will always be more than 1, that is around 2.8. So it shows what does this Hill coefficient value tell you? That hemoglobin has high cooperativity and oxygen binding in the subunits of the hemoglobin, they are interdependent, and it suggests positive cooperativity among the heme units due to heme-heme interaction. And this is a Hill point, this is a graph that is drawn to show the from this Hill equation they have drawn a graph to show you how the graph comes for Hill coefficient. Now you can see this is for myoglobin, there is no slope. Okay, the slope is unity, which indicates non-cooperativity, whereas in case of hemoglobin you can see that the slope is greater than 1. Okay, at some point it is unity. You can see here and here where f value is greater than 0.8 and less than 0.2, the slope is unity. For hemoglobin also, at this point it does not show any cooperative effect, whereas here where the slope is greater than 1, hemoglobin shows cooperative effect. And for other two, that is hemocyanin and hemerythrin, which we will be studying in the next video, they have among all these four, that is hemoglobin, myoglobin, hemocyanin and hemerythrin, hemocyanin has the greatest cooperative effect value of around 9, and hemerythrin has around 1.2, that is almost equal to myoglobin. Next, we are studying about the effect of H plus CO2 Cl minus on O2 binding interaction with Hb. Here mainly we are studying what happens in the muscles. Okay, so first let us study what is heterotropic allosteric interaction. You already studied homotrophic allosteric interaction. What is that, students? One oxygen binding to the hemoglobin favors the binding of the next oxygen atom. That is because of homotrophic allosteric interaction. Now, what is heterotrophic allosteric interaction? It is also one type of cooperative effect where binding of one atom, this time binding of one atom like H plus favors the binding of the second atom, which may be same or different. Okay, are you understanding the difference? Binding of one atom to the hemoglobin, one atom or one molecule, favors the binding of second atom or molecule, which may be same as the first atom or molecule or different. Okay, so there one oxygen got binded, next it favored the binding of second oxygen. Here one H plus will get binded and it favors the binding of second H plus or CO2 also. Okay, does not have to be H plus only, it can be some other atom or some other molecule. That is heterotropic, that is different atom or molecule. Heterotropic allosteric interaction. This is mainly seen in the muscle or the tissues of the body, and this is responsible for hemoglobin to release oxygen in the muscle and take carbon dioxide back to the lungs. Let us see how does this happen. Before that, you should know that when H plus concentration is more, the pH will be less. That is true, correct. When there is high concentration of H plus, how is the pH? pH will be less, and the oxygen binding affinity of hemoglobin is less. Why is that? Because this time the concentration of H plus is very high. Which molecule's concentration is high, that molecule will bind to the hemoglobin. There in lungs there was high concentration of oxygen, whereas now in the tissues, there is a high concentration of H plus ion. So the oxygen binding affinity of the hemoglobin is less this time. Let us see how does this actually happen. Okay, so consider your tissue. Okay, your body tissue is there. Now hemoglobin has not reached yet. Hemoglobin is still in the lungs. It is on the way coming to the tissue. What happens in the tissue? Now hemoglobin has not reached yet, so there is no oxygen in the tissue. So working tissue, it releases carbon dioxide and lactic acid. Why is this produced? Because of incomplete oxygenation of glucose. You know that in tissues there is glucose which undergoes oxygenation, but because of lack of oxygen, there is incomplete oxygenation of carbon dioxide and lactic acid because of insufficient supply of oxygen, because of which a large amount of carbon dioxide and lactic acid is released in your tissue. Okay, now lactic acid, it is acid, so there will be high concentration of H plus ions in the tissue, and how will the pH be? The pH will be very less. Now, in step number 2, the oxygenated hemoglobin has reached the tissue. Okay, this is step number 2 where the oxygen containing hemoglobin has reached the tissue, and now it sees that here in the tissue the pH is very low. Because of low pH, what happens, students? PH is less means oxygen binding affinity of the hemoglobin is less. So that oxygenated hemoglobin releases the oxygen and binds to H plus and carbon dioxide because of heterotropic allosteric interaction. Are you understanding the steps? Let me repeat what happens in the working tissue. Step number 1. Working tissue, oxygenated hemoglobin has still not reached the tissue. So in tissue, there is incomplete oxygenation of glucose because there is absence. Absolutely, oxygen is not there in the tissue. So there is incomplete oxygenation of glucose, because of which working tissue, it releases carbon dioxide and lactic acid. Because of this lactic acid, the pH of the tissue becomes very less. Now, in step number 2, hemoglobin containing oxygen has now come and reached the muscle or the tissue. Now, what that hemoglobin should do in the tissue? It has to release this oxygen. How does this releasing of oxygen takes place? Because the pH is very low in the tissue because of high accumulation of lactic acid and CO2. What happens in the tissue? Whenever the pH is less, whenever hemoglobin, oxyhemoglobin is present in an atmosphere where pH is less, the oxygen binding affinity of the hemoglobin is less. So the hemoglobin releases this oxygen and goes and binds to H plus. As soon as it binds to H plus, what happens? Step number 3. Now it has released this oxygen in the tissue, which is now taken up by myoglobin. Okay, fine. Now hemoglobin does not have oxygen in it, so it is in the deoxy form. As soon as it has released oxygen, it takes up the H plus ion. Once it binds to one H plus ion, what happens? That H plus shows heterotropic allosteric interaction, because of which it favors the binding of the next H plus or CO2. So this is the reaction that happens in the muscle. First, the HbO2 comes to the muscles having oxygen in it. From the lungs, it reaches the muscle. In the muscle, there is high concentration of H plus, because of which the hemoglobin affinity to bind to oxygen becomes less, because of which it releases the oxygen in the muscle. And next, in turn, because it is alone now, it binds to carbon dioxide and H plus because of heterosteric allosteric interaction. Okay, heterotropic allosteric interaction. So, what is root effect? The effect of pH on oxygen affinity of hemoglobin, it is described as Bohr effect. What is the effect of pH on oxygen affinity of hemoglobin? Lesser the pH, the oxygen affinity towards hemoglobin is also less. Okay. So the effect of this effect is nothing but the Bohr effect. A very large Bohr effect leading to sharp decrease of oxygen affinity with decreasing pH is nothing known as the root effect. That is, on a large decrease in the pH, that is high concentration of H plus ion, leading to sharp decrease in O2 affinity of the hemoglobin is called as a root effect. So, in what happens now, this is the complete step that has happened in the tissue. Now, the hemoglobin is now bound to carbon dioxide and H plus. This goes back to where? It goes back to the lungs. In lungs, what happens once again? There is high concentration of O2, and you know that because of high concentration of O2, whenever the pressure of O2 is high, hemoglobin gets oxygenated. That is, it shows homotrophic or allosteric interaction. In order for the hemoglobin to bind to oxygen, it should get rid of carbon dioxide and H plus. So H plus and carbon dioxide from the hemoglobin are unloaded or are released in the lungs. That is called as a Haldane effect. That is the reverse of what happened in the tissues. In tissues, it released oxygen and took up H plus and CO2. In the lungs, it is releasing H plus and CO2 and it is taking up oxygen. Okay. And one more atom that shows this type of effect is the chlorine. Chlorine also binds to deoxyhemoglobin. Thus, the oxygen affinity of that hemoglobin decreases and increases in Cl with the increase in Cl minus concentration. So, same way like H plus, if there is high concentration of Cl minus, the oxygen affinity of the oxyhemoglobin decreases and it releases the oxygen atoms. So the last one is effect of 2,3 diphosphoglycerate on oxygen affinity of hemoglobin. You already studied what is 2,3 diphosphoglycerate DPG. You know that for every one hemoglobin molecule, one molecule of DPG will be present attached to it. So this also shows heterotrophic allosteric interaction. That means that when there is high concentration of DPG, the oxygen affinity of hemoglobin decreases. That is, hemoglobin releases the oxygen and it becomes deoxyhemoglobin and it binds to DPG whenever there is a high concentration of DPG. So, in active tissues, when there is high concentration of DPG, it stimulates the release of oxygen from the oxyhemoglobin. So, in a human red blood cell, DPG, you know that it is present as the same molar concentration as that of hemoglobin. For every 1 hemoglobin unit, there is 1 DPG that is attached to it. What are the different functions of the DPG? Why is DPG required? One is that in the absence of DPG, the P50 value for hemoglobin becomes 1 torr. You know that it was 26 torr, but if DPG is not there, the P60 value becomes 1 torr. Same as that of myoglobin. But because there is DPG present in the blood, the P50 value becomes 26 torr. This value is very important. It should be high. Okay. Why? Why should it be high? Because the hemoglobin should take up oxygen in the lungs. Correct. That happens because of this P60, P50 value being high. So in case of absence of DPG, it cannot take up that oxygen. So DPG is very much required. Also, DPG maintains this equilibrium in the body. That is, number of oxyhemoglobin plus DPG is almost in equilibrium with the deoxyhemoglobin DPG complex and oxygen release. So it maintains this equilibrium in the body. Also, it is required in higher altitudes. You know that when you go to higher altitudes, like you climb mountains and all that, the oxygen pressure will be very low and your body requirement for, cannot fulfill the requirement of oxygen. That time, what does this DPG do? It binds to the oxyhemoglobin and favors the release of more number of oxygen. Whenever it binds to oxyhemoglobin, what happens to that hemoglobin? It becomes deoxy and releases oxygen, correct? So in higher altitude, when the oxygen pressure is low, DPG fulfills that requirement of oxygen of the body by binding to the oxyhemoglobin and making it deoxyhemoglobin. And also in several anemic patients who cannot, anemic patients are those who cannot fulfill the body oxygen demand. Okay. So in such patients also the DPG concentration increases in order to meet the body oxygen demand. And also it is found that the DPG binds more strongly in adult hemoglobin compared to that of the fetal hemoglobin. So if there is a baby in the mother's womb, To the mother's hemoglobin, the DPG will bind very strongly, whereas to that baby's hemoglobin, the DPG won't bind that strongly. Because of that, what happens when it binds, that DPG binds strongly to the mother's hemoglobin, there is release of oxygen from that hemoglobin, and that oxygen will go and bind to the fetus hemoglobin. This is how there is transfer whenever a baby is growing in the mother's womb. Where will that baby get oxygen from its mother? Correct. So this is how it happens because of DPG. That happens, that is, it helps to deliver that oxygen from mother to the baby. And also in a condition of hypoxia, where there is disorder, same like anemic patients only, they cannot meet oxygen requirement, DPG is required in order to fulfill their oxygen requirement. Next part is we studied completely about hemoglobin, myoglobin, what are the different effects they show. So a small comparison, what is the difference between hemoglobin and myoglobin? All the things that you've studied so far, you will put it in a list and write. So hemoglobin is tetrameric, whereas myoglobin is monomeric. Its molecular weight is 64 kilodalton, whereas in myoglobin it is only 16 kilodalton. And this acts as a carrier protein, whereas this is a storage protein, and it shows cooperative effect, whereas myoglobin does not show any such cooperative effect. And hemoglobin, other than oxygen, it can bind to H plus, DPG, carbon dioxide, Cl minus, whereas myoglobin does not bind to any other atom other than oxygen. And Hill coefficient value is high, that is around 2.8, whereas that for myoglobin is 1. And P50, that is percentage oxygenation, is also high, that is 26 torr for hemoglobin, whereas for myoglobin it is only 1 torr. And the last part is the function of the globin protein. What are the functions? One is it shows allosteric effects, and also it protects protection of hemoglobin and myoglobin from irreversible oxidation. Then you know that sometimes hemoglobin and myoglobin can suffer CO poisoning, but this is prevented by this globin chain. It also shows sigma donor properties, and it also helps in protonation and deprotonation of the globin chain, that is important for maintaining the biological pH and CO2 transport. So with this, we have completely covered the topic, that is hemoglobin and myoglobin. I hope you have understood this. Thank you for attending my class, students. Take care. Bye.